The last several videos covered transcription and translation, taking DNA and turning that DNA into a protein. And while every cell in your body has the same DNA, not every cell makes the same protein. So how does a cell determine what genes are going to be transcribed and what genes are not going to be transcribed? This schematic you see on the slide is one example of regulation of DNA transcription. If a cell has enough tryptophan, tryptophan is an amino acid that's used to make proteins, that tryptophan is going to inhibit the production of more tryptophan. One way to do that is to inhibit the production of this tryptophan production gene. If we stop transcribing this tryptophan production gene, we're not going to make the enzyme that's used to make tryptophan, and we'll reduce the amount of tryptophan that is being produced. So this is an example of negative feedback where the product of this process is going to regulate the production of that enzyme by regulating gene expression. There are several different mechanisms of gene regulation, and I'm just going to talk about two of them. I'm going to give one example that includes negative regulation and an example that gives positive regulation or demonstrates positive regulation of gene expression. The most common example of negative regulation is the lac operon. The lac operon is a group of genes. This lac operon is found in prokaryotic cells, but the lac operon includes the genes LAC-Z, LAC-Y, and LAC-A. This gene LAC-Z is going to make the enzyme beta-galactosidase, which breaks down the sugar lactose, so it makes a lactase enzyme. The lac operon also includes a region that is called the operator. This is the operator. The operator is going to bind a protein called a repressor. The repressor is coded for by this gene called the LAC-I gene, which is a regulatory gene. So it's going to produce this repressor, which is going to regulate the production of the genes that are part of the lac operon. Now, the lac operon also includes a promoter. That promoter is where our RNA enzyme, our RNA polymerase enzyme, is going to bind in order to cause transcription. Without lactose present, the repressor is going to bind to the operator. So if there's no lactose, our repressor will bind the activator, and we are not going to make lactase genes. Your cell doesn't need to make lactase genes if there's no lactose to digest, so we're going to repress the expression of that lac operon. However, when lactose is present, that lactose is going to bind to the repressor, making that repressor inactive. Now that the repressor is inactive, RNA polymerase is free to bind to the promoter, it's free to transcribe our lac operon, it's free to make the enzymes such as beta-galactosidase, which is a lactase and which will break down the sugar lactose. This is an example of what we call an inducible operon. The signal molecule turns on gene transcription. So the transcription of the lac operon is induced by the signal molecule. The signal molecule in this case is this allolactose, so a form of the sugar lactose. These inducible operons are usually in metabolic pathways. You only want to make enzymes when the nutrients are present. If you don't need the enzyme, don't make the enzyme. If you don't need, in this example, beta-galactosidase, don't make the beta-galactosidase. So you're only going to turn on that gene when the inducer, in this case lactose, is present. This is also an example of negative regulation. It's negative regulation because the presence of an active repressor stops transcription. There's a protein interaction, or the protein interacts directly with the DNA and suppresses the transcription of that DNA. I'm now going to make this lac operon a little bit more complicated. Right? The presence of lactose only induces transcription by inactivating the repressor. The cell only wants to use lactose when glucose is not present. Glucose is a better source of energy than lactose, and so the cell's first choice is going to be use glucose as an energy source. Its second choice is going to be lactose. So now we have to address another section of the lac operon, the CAP binding site. When we have low levels of glucose in the cell, we have high levels of cyclic AMP. These high levels of cyclic AMP are going to activate the CAP protein. CAP stands for the catabolite activator protein. CAP is an activator. It's a protein that's going to bind to our DNA and stimulate gene transcription. So our cyclic AMP is going to activate our CAP. It binds to an inactive CAP, thereby activating CAP. The CAP then binds to the DNA, and this is going to increase the affinity of RNA polymerase for the promoter. So it's going to increase the amount of RNA polymerase activity. Okay. So let's look at this from the big picture, looking at both the role of the repressor and the role of this CAP. Right. If lactose levels are high and glucose levels are high, we are going to have an inactive LAC repressor because the lactose is going to bind to that LAC repressor and it frees up the operator. So RNA polymerase can bind, but it doesn't bind very efficiently because the CAP is inactive. We're only going to have a binding of RNA polymerase, so we're only going to have efficient binding of RNA polymerase when there is an active CAP. So when there's low levels of glucose, so high levels of cyclic AMP is going to cause CAP to bind. That's going to cause the RNA polymerase to bind. RNA polymerase can only then proceed down the operon to transcribe our LAC-Z gene if the repressor is inactive. So this role of CAP is an example of positive regulation. The CAP is a protein that's going to interact with the DNA to increase transcription. So the LAC operon is an example of gene regulation in prokaryotic cells. Eukaryotic gene regulation is much more complicated, and we're only going to touch on a couple concepts. We're going to talk about gene regulation at the DNA level, at the transcription level, at the post-transcription level, and we'll start here with the DNA level, where we have acetylation and methylation affecting DNA regulation. One mechanism of DNA regulation is histone acetylation. An acetyl group, this molecule over here, this functional group over here, is added to our histones. If you recall, our DNA is packed as chromatin. The DNA is wrapped around histones. Those histones form a nucleosome, and those nucleosomes are packed together to form our dense chromatin structure. By adding an acetyl group to the histones, that opens up our DNA structure. By opening up that DNA structure, by allowing the transcription enzymes access to that DNA, we can now transcribe that DNA. In contrast to acetylation, DNA methylation is going to prevent transcription. We have methyl groups that are going to be attached to our DNA, and that is going to prevent our DNA from being transcribed. So DNA regulation occurs through acetylation and methylation by controlling how much DNA is available for transcription. DNA is also regulated at the transcription level. We have both what are called proximal and distal control mechanisms to regulate transcription. Distal and proximal control elements are going to facilitate the binding of the transcription initiation complex. Right, if you recall that during transcription, RNA polymerase II is going to bind to our promoter. We have that TATA box in the promoter, and that is where our RNA polymerase II is going to bind. These proximal control elements, which are near the promoter, and these distal control elements, which are far away from the promoter, are going to affect the binding of transcription initiation complex, which includes our RNA polymerase II. These groups of distal control elements are called enhancers. These enhancers, again, aid the binding of transcription factors, and these enhancers can be thousands of nucleotides upstream or downstream of the TATA box. They can even sometimes be found within the introns of our DNA that's going to be transcribed. Our proximal control elements are adjacent to the promoter, and we see molecules such as repressors that can bind to these same regions. So these proximal control elements can have molecules that bind to facilitate transcription. They might also have repressors that bind to suppress transcription. There are additionally some post-transcriptional ways that we can regulate our protein expression after we have made our mRNA. One way we can regulate protein expression is through alternative splicing. The same mRNA can be cut multiple ways to make different proteins. So here on the right is a schematic of a section of DNA, and that section of DNA is transcribed into a primary RNA transcript or pre-mRNA, and that mRNA can be spliced differently to make different proteins. In this example on the left, we leave out the exon number 4. On this example on the right, we leave out exon number 3. And so these two mRNAs are going to make different proteins. So we can control our protein expression through alternative splicing. We also control protein expression through mRNA degradation. Once we produce an mRNA, it can be translated repeatedly until it is degraded. By controlling the breakdown of mRNA, you can control how much protein is produced. If you need a lot of protein, you don't want to break down your mRNA very quickly. If you only need a little bit of protein, then that mRNA is broken down rapidly, so only a little bit of that mRNA is translated into proteins. Protein levels are also regulated after the protein has been produced through differential processing. The protein regulation can occur through processing or packaging or shipping or degradation. Proteins are processed and shipped by the Golgi apparatus, and the Golgi can control how they are processed. Those proteins often need to be cleaved and assembled into functional proteins from several subunits, and so that assembly, regulation of the assembly of proteins can affect the regulation of protein function. We've already talked about an example of protein degradation, or I should say the regulation of protein activity using protein degradation. That example was with cyclin. Cyclin, if you recall, fluctuates during the cell cycle. As cyclin levels increase, it can bind with cyclin-dependent kinase to get the cell past the G2 checkpoint by forming maturation promoting factor, and then the cell will proceed through mitosis. After mitosis, that cyclin is degraded, thereby deactivating the maturation promoting factor. And so we regulate the activity of this protein cyclin-dependent kinase by regulating the degradation or production of this protein cyclin. Proteins that need to be broken down are marked by adding a small protein called ubiquitin. So the cell uses the presence of this protein ubiquitin to recognize proteins that need to be degraded.